Okay, so I'm Lisa Cubedo. Did anyone do functional proteins and genes last semester? Okay, so you will have seen me last semester. For the people that haven't, hi, I'm Lisa, and I'm the coordinator for molecular biology. I will be on maternity leave a little bit later on in the semester, but it shouldn't affect how this unit runs at all, and I'll go through briefly the slight differences we have between this semester and how we've run the unit because of this. Okay, so just a very short background, given now it's already 10 past. So I coordinate molecular biology, and it's generally my area of research as well. So our research lab looks at proteins that are involved in DNA repair. What you'll see when you look through the lecture material, the end of the lectures, you have two lectures on DNA repair because it's very important for the DNA processing and making sure we uphold our genetic integrity, which we'll be talking about throughout this unit. We also work on proteins that remodel our DNA so it can get accessed to turn on expression of genes, which we'll be talking about as well, and things like that. And the pictures are essentially after I did a PhD at Macquarie Uni, and then after I did a PhD, I went to work in a DNA repair lab in the UK. It was actually in Scotland, and which is the top thing. That's St Andrews in Scotland. And then I came back to Sydney Uni, which I was there for about 7 or 8 years, and I had a research lab, and we were working on DNA repair, and clearly now I'm at Western Sydney. I'm actually based since February at Parramatta, at the Parramatta campus, so the best way, and people will have already been in contact with me by email, but the best way to contact me, especially also during the semester, given I will not be answering my work phone in about a month or so, clearly, email me, okay? So an email is the best way to contact me, and if you have already contacted me, you'll see that I actually answer my emails. So that's essentially the best way to get a response from me quite quickly. And I did have, in your notes, I think I did have an extension. That was actually my Campbelltown extension, so I forgot to remove it. So email is the best way to contact me. Okay, so the way that I'm going to run the unit this semester is slightly different than last. So the beginning actually nearly all of the unit for lectures runs the same way. So I always do the first block of 4 lectures at every campus, and then my colleague, Dr. Mark Temple, always does the next block of 6 lectures at every campus. And then I usually come back and do the last block of 3 lectures at every campus. So the last block of 3 lectures I won't be doing. My colleague Roland Gamsegger will be doing it, but that's still fine because he actually teaches the whole unit in summer, so he's very engaged with the molecular biology unit. So all 3 people have continue to lecture and teach into this unit, so there's actually no one new. Okay, so what will be actually, so nothing to do with me going on maternity leave. Unfortunately, what happened with timetabling this semester is they stuffed up timetabling quite well. Mark Temple runs a 3rd year unit as well, and his 3rd year unit runs on 3 campuses, and he teaches everything in that 3rd year unit. He also teaches in this unit on 3 campuses, and unfortunately, timetabling, you have to put this stuff in at the beginning when you're requesting, because then you get this timetable gets spat out, and then you can really not change much. And unfortunately, what happened this time was that every lecture that they've allocated for molecular biology, Mark had something on at another campus for his unit, and we couldn't find any contingency that he could actually give a lecture and do his unit at the same time, which it's quite unfortunate, and doesn't usually happen. They usually take these things into account before they do the first draft of the timetable, but they didn't this time, and so there was really nothing that I could do. So what Mark will be doing, and he does this stuff really well, is he's going to be posting recorded lectures. So not lectures that have been recorded via these lecture recordings, but his own recorded lectures where you can see him in the corner talking as well. So he does this stuff really well. He's done it for his own units as well. So this block of 6 lectures, you won't come into you can come into the lecture theatre, but Mark will not be present in the lecture theatre. So it won't be a face-to-face lecture. These lectures 5 to 10. So there's a block of 6 lectures that Mark won't be able to be present. He's probably going to be at this campus, but he won't be present to give this lecture because he'll be teaching in his own unit. So he'll post those lectures clearly before the lecture time, and they'll be in the same lecture folder where you would access the PDFs of the lectures, of my lectures and Roland's. They'll just be his own folder, and in there'll be his own recording that he will have done, and he usually does them in his office. They're done really well, okay? So, and they're quite interactive. So please give feedback. So clearly this is not normally how we run the lectures in this unit, to do these online lectures, but that's how it has turned out this semester, and we won't be able to change that, unfortunately. So you can give feedback at the end of the semester and let us know what it is, how it is that how you felt about those ones, but Mark won't be able to be at the lectures in person. But you'll see him talking, because he has a little picture and he's talking while he's giving his online lecture. So he's really good at these things. Okay, so let's continue. Right, so the textbook is in the library, so you don't have to buy it. The textbook that we're using is called Molecular Biology, and we don't do the whole textbook. We do parts of the textbook, and essentially I tell you at the beginning of the lecture what bits of the textbook we'll be covering, but you'll then see them if we're using figures from those chapters. You'll see which bits of the chapter it is that we're covering. So it might be that the textbook's a really good way to revise what we've done in the lectures. Just keep in mind that we won't have done the whole of a chapter, okay? So we may have done sections of a chapter, and you have to take that into account if you look at the textbook. So you can also get online version now of this molecular biology textbook, so have a bit of a look and see if that interests you. But as I said, the textbook's also in the library. I like this textbook because it's got a lot of protein structures in there, and one of the reasons why it has a lot of protein structures is because one of the authors, Jennifer Doudna, is a structural biologist, and they have incorporated a lot of knowledge of how all of these DNA processing events that we're going to talk about work, is because we know the structures, the 3-dimensional structures of the proteins involved, and we can then determine how they actually work biochemically. So they've incorporated a lot of that into this textbook, and for me, I think it's a really useful way of understanding these DNA processing events that we're going to talk about. Okay, so it's not a small amount to learn. So remember, you're a second year, so you're going to, essentially you're not first years anymore, you're not first semester, second year, and in fact, by the end of this semester, you'll only have 1 year left, where after that you'll potentially go and get a job, okay? So we're assuming that you're putting in effort for yourself to do well in units, and to show a bit of responsibility and actually study for units if you actually want to go well in them. Okay, so I generally put this most frequently asked questions up because they are the most frequently asked questions from students. So first one is always, what's going to be in the final exam? Everything, so all the content of the lectures is examinable, and the second question is related. Will anything from this computer class that you will have already been to, or the practical classes, will they be in the final exam? So yes, because you have to know the concepts that we're doing in the practicals. What you'll see, what we're doing in the practicals and what you started doing in the computer prac is cloning. We're going to talk about cloning in the lectures, and you're going to do this whole process of cloning during your lab modules. So yes, it's not some strange thing that you're doing on the side there. It's an integral part of understanding molecular biology, is what you're learning in the practicals. And you need to know how to do specific calculations for these practicals, and you will be also you'll be also examined on these basic calculation that everyone really needs to know how to do if you're going to work in a lab, and someone is going to think you know how to do these calculations at the end of your bachelor degree, if you go and work in a lab, for example. So, for example, making a buffer. Everyone has to know how to do that. So there's no way that someone's not going to think that you know how to do that after you've been in a 3-year degree, and especially have done a unit like molecular biology. Okay, so the final exam is the current format is a multiple choice exam, but as I said, it does have calculations in there, even though it's a multiple choice exam. Okay, so as you've seen, the first bit of this lecture is looking at the breakup of how the unit is run, and then we're actually going to start doing an introductory lecture. Okay, so this slide just shows you a summary of the assessments that are part of this unit. So all of this information, don't have to write it down, it's in the learning guide. Okay, so I've posted the learning guide up on Views, and this information is replicated in there. Okay, so the learning guide, at least for my unit, is very detailed. You should find essentially all the information that you require to complete your assignments, due dates, how much things are worth, how you're going to be marked, marking criteria. It's all in there, okay? So I've spent quite a bit of time working on that learning guide to make sure this information is in there, so you can access that, and you should be accessing that before asking me when is the due date for something. Okay, so you can see the assessment items are broken up into 3 things. You can see the first item is a quiz, and you will, if you've seen, if I was in here just before, so everyone seems to do a quiz in the middle of the semester. Okay, so I haven't called it mid-semester quiz. It's an online quiz. It's in the middle of the semester. Okay, so the point of this quiz is, again, as all unit coordinators want to make sure that you're tracking well. Okay, or that if you're not, that you know what it is that you don't know, essentially. So after you do this kind of quiz, then you'll see, you know, which areas that you need to concentrate on. If you still don't know how to do calculations, because you will be, again, you will have calculations to do in this online quiz in the middle of the semester. But that's the point of having a quiz that is not at the end, clearly, that you see how you're going, see how you're tracking. So you can see it's not worth a small amount, but clearly if you don't go well, it's not the end of the world. You can still go well overall. Okay, so the second bit of this assessment is where there's quite a few parts to it. Okay, so it's the assessment item that has to do with the laboratory practical and having a bit of a look at understanding the polymerase chain reaction, which is a crucial tool that we use in molecular biology. You're going to be using it during your lab modules, and we're going to be talking about it in the lectures. So it's a crucial method that you need to know how it works and be able to explain that. So that's one of the parts of this laboratory and practical component assessment is your written assignment, and we'll talk about this PCR critical analysis in the next slide. You also have quizzes to do. So you didn't have a pre-lab quiz for your computer lab. You came straight in and did that. I know you didn't have a lecture before your computer lab, but it wouldn't have made any difference to starting to understand the process of cloning, which is what your computer lab was about, because that's not what we're talking about in this introductory lecture today anyway. So it's not that you missed out on something that you should have, that you needed to do your computer prac. So the computer prac was essentially the paper version of what you're going to do in your lab modules. Okay, so for each of your lab modules, so the ones that you go into the wet lab, what we generally call it, you do a pre-lab quiz, and that's essentially to make sure that you're reading the lab manual before you come in, and that you do or attempt any of the calculations that are required that you need to have done before coming into the prac. And they're calculations like you'll see once you look at the lab manual, once you set up a polymerase chain reaction, to do some dilutions from stock solutions. You're going to have to know how to do those. You will be making up the buffers. You're going to have to do that. So simple mole calculations. I know everyone's done them, even though you may not remember. Everyone's done chemistry, and you did mole calculations in chemistry. So I know you've done them. I've seen that. So if you can't remember how to do them, you can go back and look at your chemistry, because it's not a little compartment filed away over there. You actually build on knowledge that you've gained in first year, and then you use it again in different units, okay? So just because it was chemistry doesn't mean it's not related to any other unit that you're doing. Okay, so, and if you really don't remember how to do any of these calculations, as your lab supervisor will be happy to go through in little groups and make sure that everyone knows how to do these calculations. So you can't get away from being examined on how to do these simple dilutions, simple mole calculations. They're not complicated, but you do have to know how to do them, and you will be assessed on them various times, okay, in the pre-lab quiz, in the mid-semester quiz, and in the final exam, okay, so you can't get away from that. Before your lab. So it opens a week before, and it closes at the time that your lab is on. Okay, so so you've got this 10% of this. So you can see that this laboratory practical component is a big chunk of your mark in total. Like, so it's nearly half of your mark, and this is to really stress the point that understanding what you're doing in the practical is really important concept, and we want to make sure that you know how to do all of these things, calculations that you need to know how to do, understanding the methods of cloning, and all of these other processes that we're going to do in the lab in the laboratory. Okay, then you get another 10% of your mark is your in-class marks. Yeah. They're not unlimited times, but they're not many questions, so you have plenty of time in there for those. So they're not unlimited, but you will definitely have plenty of time that I've allocated to do those pre-lab quizzes. Say that again? Yes, they're only one attempt. Yeah. Okay. So then there'll be some in-class marks. So we want to make sure that you understanding what you're doing in your practicals. So the in-class marks in the computer lab, you can see that you were marked on, whether you were able to produce these paper cloning versions of plasmids with your gene inserted in there, and whether you were able to explain how you got those calculations. For the other practicals, you're essentially going to be marked on your results. Okay, so you're going to be producing. You're going to be doing some polymerase chain reactions, different types. Then you're going to be running them on a DNA gel. Then you're going to have to tell us what's on the gel. Okay, so you're going to have to do some explaining, because everyone has to image their gel. So you will all have an opportunity to be tested face to face what it is on your gel, and if you can explain what that is. And based on that, you'll get a mark, an in-class mark for each of your lab modules. And then the final thing, we don't collect lab books in this unit. We look at the lab books, and we'll go through that in a little bit more detail. You have what's called a final prac report, and I'll just leave that for the I have an extra slide about that. And then there's obviously the final exam, and it does have a threshold. You must get at least 40% in the exam to pass the unit. So there have been people, students over the years that get you know, they passed the unit, they might even have a credit 60%, but they didn't get 40% in the exam, so they get a compulsory fail, okay? So there's no leeway here. Doesn't matter how well you did in the other component. If you can't get 40%, which means you don't know half of the stuff, then you're not going to pass the unit, okay? Because that will not satisfy the requirements that you know more than 40% of the knowledge that you require in this unit. Okay, so the online quiz, I won't go in more detail anymore. It does, when you look in your, so this, again, all this is in your learning guide. You don't have to copy any of this down. So it tells you up to which lecture that you have to, that you'll be examined on. And it's pretty much up to when we talk about RNA synthesis. And that's the start of Mark's lectures. Okay, so this is, so the PCR critical analysis is part of this lab component, this overall big chunk of your Mark, which is this lab component, is your written assessment. So the whole point of this PCR critical analysis is it's broken up into two parts. First part, you're explaining what PCR is. There's various things that you need to explain within that, and they're detailed in the in the learning guide in this section. So you can see that it's detailed overview of how the polymerase chain reaction works, because it's an integral method that we use in the molecular biology lab. And so you can see there's a few things that you have to address. And then the second half of this assessment, which I don't appear to have in here, is, don't know where the other part of it has gone there. The other half of it is that you have to find a method, or that you have to decide on a particular method that uses the polymerase chain reaction. So it might be there's a lot more forensic students here at Hawkesbury, and generally a lot of students kind of pick that. So how you would use the polymerase chain reaction in crime scene investigation, things like that. You can do some biomedical method that uses the polymerase chain reaction, etc. So there are these two parts that forms part of this whole PCR critical analysis assessment. Okay, so the laboratory assessment, what you're being assessed on actually in the lab. So it's pre-lab quizzes, and then we have a look at your lab book. So we want to see that you're actually writing something, but we're not collecting it at the end. And that will be done within the lab, within the prac time. So we do want you to, for all your wet lab practicals, to prepare a flow chart of what it is that you're going to do. So very basic, simple flow chart of the main steps that you're going to be doing in that lab module. So the other thing that I should reiterate, if you haven't gotten that already from your computer lab, is so your computer lab is basically what you're going to do in the lab. Okay, so it's a paper version of the cloning that you're going to be completing in these 4 lab modules that you'll do in the lab. Okay, so they're not little separate practicals. One practical, the stuff that you make in that prac, so you're going to do a polymerase chain reaction, whatever it is that you're going to do, that material that you prepare in the first lab module, you're going to use it in the second lab module. You can do something with that, then you're going to get some more stuff, and then you're going to use that in your third lab module, and then you're going to use that in the fourth lab module. Okay, so they're continuous. That's why you need to go to the same prac. Please don't shift around pracs, because your material is your material. Okay, so if you don't have any material, too bad. If you stuff up, we generally have contingency plans for if you stuff up and you don't have material to continue, you still have to do the practical. But the idea is that this whole cloning process, so amplifying up this bit of the TP63 gene that you did paper version yesterday with those 2 oligonucleotide primers, introducing a mutation, putting it into this plasmid, transforming it into bacterium, making a plasmid DNA and checking with BTS1 that you've got the right thing. That's going to happen across these 4 lab modules, okay? We usually do that in the lab in maybe 2 days or something like that, but you'll take 4 lab modules to complete the same thing. Okay, so the final prac report is essentially what you're going to, is a prac report on all 4 lab modules, okay? So the results that you've gained doing this whole cloning process. So it's very detailed in your learning guide, what you have to do for your final prac report. We also have a prac tutorial after your 4th lab module that goes into a lot of detail of what's expected of you for the final prac report. So you should have a very good idea by the end of that module 5 what it is you need to do for that final prac report. And that's essentially the DNA gels that the results that you get in each lab module. You have to discuss them, okay? So you've got to present you've got to give us some aims. What is it that you're supposed to have done in these 4 lab modules? Present your results, so pretty much your DNA gels, and then discuss them, okay? So that's the basis of your final prac report. And you can see it's worth 15%, so it's quite a big chunk. Okay, does anyone have any questions now about anything that I've talked about? No? Okay, so as I said, all this information is in your learning guide. I generally send quite a few emails during the semester and you obviously, or announcements, but that you get them also as emails to your Western Sydney account. So if you don't check your Western Sydney account, you don't see the emails, okay? So you're going to have to check, even though that may not be your primary email account, you're going to have to check that, because that's really the only way that we can contact you or send you information. And I do send quite a bit of information during the semester, and especially when, for example, things are due, things are coming up, what's coming up. So there's really not going to be a huge excuse to say you didn't know something was coming up, because I generally tell you with enough time that something is going to come, that you're going to hand in something, or that a quiz is coming up, etc., okay? But you have to read your email to see that, okay? Okay, so with that, we're going to actually start the intro lecture today. So the whole point of this introductory lecture is just to give you a bit of a sneak peek on the things that we're going to be covering in a lot more detail throughout all the lectures. And yeah, so it's kind of a more, an introduction on the small bits that we're going to be looking at in more detail in either my lectures or Mark's lectures throughout the semester. Okay, so the lectures are broken up into themes. So the first theme that we're going to be looking at is working with DNA and RNA, because DNA is essentially the storage form of all our genetic information, and what you'll see is we need to convert DNA into RNA to finally make a protein from that gene that is stored in our chromosomes. Okay, so as you can see here, I've listed a set of chapters from the molecular biology textbook that you can have a look at, that we have a bit of a look at in this lecture. Okay. Then I usually set up, do a bullet points learning outcomes for my lectures, and that's simply to remind you well, you can use it as a revision tool, but it's to tell you exactly what we're going to cover in this lecture, and you can use it as a revision tool that you can actually use those points and say, okay, do I know how to do I know all these points, or how to describe all these points? So I won't go through them in detail because that's exactly what we're going to do in the lecture now. Okay, so an interesting question always comes up is, you know, molecular biology is very central to many fields, biomedical fields, you know, genetics, immunology, but also forensic science, plant biology. If you think of all the genetically modified crops that there are now, molecular biology is the way to make those crops or those plants. So it is central to a lot of different fields in biology. So the idea is that molecular biology, it's not so much a technique, it is a technique. So there's lots of tools in the toolkit of a molecular biologist, but I think the idea is really the fact that for us to be able to make these new techniques in molecular biology or develop new techniques, we need to understand how a cell works and how the information gets processed from this storage form of DNA, that where all our genes are stored, if we're humans, in our chromosomes, to actually making a final protein product from that gene, and the protein is the product that actually does the stuff in the cell. Okay, so there's, you know, if you've just eaten a donut, you're going to have to break that down. So there's going to be some cellular signal to say, oh, we need all of the enzymes, proteins, that are in the metabolic pathway to break down glucose, and we need to make those. Okay, so there needs to be a signal to say, okay, we've got to turn these genes on inside our chromosomes. How do we do that? Okay, so we're going to be talking about this information processing to get your final protein that actually does what it's supposed to, what is required of it in the cell. Okay, and so the idea is that that for us to understand, for us to develop new technologies in molecular biology, we really need to understand how cells work and the DNA processing events that occur. You can see that there, just topically, there are quite a few journals. So where research groups submit their research papers that are directly related to molecular biology. So you can see one of the Nature journals is really popular, Nature Structure and Molecular Biology, or Journal of Molecular Biology. Both of these are very good journals, very well ranked journals. And you can see the kind of research areas that the research papers that scientists submit to these journals are, and you can see really talking about structure and function of proteins and nucleic acids, repair, DNA replication, transcription. Pretty much lots of the things that we're going to talk about in this unit, they're the kind of things that get covered in these types of journals. Okay, so just to put it into perspective, this field of molecular biology, although it did exist before the polymerase chain reaction was invented, the polymerase chain reaction, or PCR, was really a turning point in the molecular biology field. It really kind of started this era, this true era of molecular biology. And so it was really not that long ago. It was only 1985. So I was small when PCR was invented. So for me, it doesn't seem to be that long ago. And the whole point of being able to, the invention of the PCR reaction was you can make millions of copies of DNA from a very small, scarce sample of DNA. And we now use this as a routine tool in molecular biology, but the idea is that before you couldn't do this. You couldn't, there were so many things that you couldn't do because we didn't have the tools yet to understand how to do the polymerase chain reaction. And the only reason why we can use the PCR reaction as a tool is because we understand how the DNA processing events occur in cells. And I'll put the PCR reaction into context when we talk about it in I can't remember, 1 or 2 lectures' time, because it's essentially a process that occurs when we replicate our DNA in cells, and we've taken that little process of replicating DNA and use that as a tool in molecular biology, and it really, as I said, heralded a new era of molecular biology. And it's not that there are not new tools in the molecular biology toolkit. There's always something new coming up, and one of the main, I guess, one of the main revolutionary processes or tools that have been invented more recently is this CRISPR system. And this is essentially the way, it's a very exciting procedure to be able to use, and you can edit genomes. So you can efficiently and reliably edit one particular gene in a genome. And that kind of this discovery of this CRISPR system was only around 2012, 2013, but the people that discovered this CRISPR system will eventually get a Nobel Prize. And one of the people is that Jennifer Doudna, which is one of the authors of that textbook. So it was really a kind of a revolutionary tool that, although has a lot of promise to be able to use it, for example, to correct mutations in humans, for example, in an embryo, etc. So it has it's a very exciting tool, genome editing tool that has only come about more recently. And again, the nucleases that usually perform this process in a bacterium. Again, the only reason why you can use it as a tool now is because you understand how this process works in the organism that it came from. Okay, so what we're going to, one of the main thrusts of molecular biology is to understand this whole information processing pathway. Okay, so DNA, so our storage form of all of our genetic information, is stored as DNA. Okay, so nothing can happen. Nothing happens to DNA until there's a cellular signal to say, you know, either you need to replicate your genome, or you need to turn expression on of a particular gene, or you need to repair that DNA because it's been damaged, etc. But the whole process to actually make a final protein that does something in cells, you have to go through this whole information processing pathway. And so while we're talking about this information processing pathway, we're also going to be talking about the idea that the information that we have from DNA is a really powerful thing. And once we have a lot of information from different genomes of different organisms, or lots of different humans, for example, we can start having a look at similarities and differences, or evolutionarily important genes that are clearly important for a specific function in the cell. And so we'll be looking at this in the next few lectures. Okay, and so the idea is why we need to understand it is we need to be able, again, this idea of to be able to apply these tools of molecular biology. So now we use them as tools. We really need to understand the molecular basis of how cells work. Okay, so the organisation of cells, how we express genes, etc. And then the powerful thing is to be able to understand similarities, or to be able to see similarities and differences in the molecular biology of different organisms. And that really has only come about because we can actually now get sequencing for genomes. Okay, so before we were, with the advent of the polymerase chain reaction, we were able to do a kind of a more rudimentary DNA sequencing. It's called Sanger sequencing, and we're going to talk about it in my lectures. And that sequencing, it's just a modified version of the polymerase chain reaction, what you'll see when we talk about it. But that sequencing was slightly limited in the sense that you couldn't sequence very long genomes. So you could sequence, we had a lot of, before 2001, where the human genome was sequenced, we had a lot of sequences of genomes from bacteria, kind of smaller organisms that you could actually use that type of Sanger sequencing to sequence smaller amounts of DNA. So it really kind of put a dampener on sequencing larger genomes. And in fact, only in 2001 was the first iteration of the human genome the sequence, and that really was a really big push from hundreds, if not thousands, of people, of researchers, to put together a first published sequence of the human genome, and you can see that was a Nature paper. In this time period, so it was not the first version of the human genome was not complete. It was nowhere near complete, but you had an idea of the majority of the genes that were in the human genome. You can see that between 2001 and 2010, there was a 10-year gap. And within this 10-year gap, there was a new way of DNA sequencing developed, so another molecular biology tool. And it's called next generation sequencing. Oh, sorry, I thought you... so it's called next generation sequencing, and it really allowed this new modified version of the normal DNA sequencing allowed much cheaper, less labour-intensive ways of sequencing genomes. And in fact, you can go and sequence your own genome now. Cost a couple of $1,000s, and you'll get all the information that you require if you want to know whether you've got a defect in some gene. Unfortunately, you will get all the information that you may or may not want, but now it's so much easier and cheaper and quicker to sequence genomes of large things. So a human genome is the largest organism, and you can do that quite quickly and easily now using this method. And within this 10-year time span, this method, this new way of DNA sequencing was developed. And you can see in the next Nature iteration of the Human Genome Project, you can see that there was now sequences. While this was a sequence of one human genome, this is now sequences of 2,000 individuals from different populations. And once you start getting information like that, you can start to see, use the power of the information that you get from a genome to see what similarities and differences there are between even different populations in the same type of organism. Not to mention then looking at the similarities and differences between, say, for example, humans and bacteria or whatever, plants, or etc. Okay, so I've been talking about genome, and let's just make sure that we are clear on the kind of nomenclature that we're using. So the DNA of an organism is called its genome. Okay, so all the DNA present in a particular organism is called its genome. And you can see on the top thing, the top rank there, the human is the largest genome. We've got about 3 billion base pairs. We have chromosomes, and that's a eukaryotic feature. And you can see we've got a certain number of genes that will then make us that certain number of proteins. Okay, so remember, a gene is always going to be made into a functional protein that does something in the cell. And you can see when you go down the list of other eukaryotic organisms, they also have quite a few base pairs of DNA, also a certain number of genes. And even when you start going down to a prokaryotic cell, so a bacterium, you can see there's still quite a few genes that are required for that bacterium to work, etc. And once you have sequences, which we do have of lots of different organisms now, you can start to really compare and contrast the genetic information that there is in those genomes, and we'll be talking about that in subsequent lectures. Okay, so what I want to actually, yeah. Oh, let me just go through this and then we'll have a little bit of a break. So what I want to introduce here is this, is to get an introduction on this information flow that we're going to be that Mark actually will be concentrating on quite a bit in his block of 6 lectures. This idea that DNA needs to be made into an intermediary form, which is RNA, that then can finally get made into a protein. So if we're talking about a eukaryotic cell, remember we're talking about a cell that has compartments, where the DNA is going to be in the nuclear compartment, but all of the other making of the protein from that particular gene happens in the cytoplasm. So we have to get that RNA out of the nucleus and into the cytoplasm to be able to make that protein from that gene. So the information flow in bacteria is much more, or the information flow pathway is much simpler in bacteria because we don't have compartments to deal with. So what Mark will be dealing with is, and what I'll introduce here in these next few slides, is this process of going from DNA to RNA, which we call transcription, and then the process going from RNA to the protein, which is called translation. And we'll briefly have a bit of a talk about that. And you can see in bacteria, this whole process is continuous, and it happens within the same area of the cell. There are no compartments, so you get DNA or a gene being transcribed into RNA, being quickly made into a protein. While in eukaryotes we have a process that's much more complex, where we have transcription, so making DNA into RNA happens in the nucleus. We then have to export that RNA, and then we can finally make protein from that RNA, and that happens in the cytoplasm of eukaryotic cells. Okay, so I think we'll take a 10-ish minute break, and then I'll continue in the next slides. Okay, I think we might start again. I think it was around about this time that I said. Anyway, okay, so the next few slides I'm going to introduce this idea of the fact that DNA carries information to synthesize RNA, which is the intermediary molecule in this whole information processing pathway, and then RNA carries the information to synthesize a functional protein, which is what does most of the things in our cells. So that the type of RNA that carries the information required to make a protein is called messenger RNA. So it carries the message of what the protein is supposed to look like, and it's often called mRNA, but its full name is messenger RNA. Okay, so let's have a bit of a look at what happens here. So the idea is that the DNA is essentially just your storage form of your genetic information. And we need to make this intermediary molecule, which is called RNA. And we're going to have a look at the building blocks, the differences between DNA and RNA in a few slides. So I think the whole process of transcription, you can think about it as a copying method. So when you transcribe something, you're essentially copying something down, okay? And that's the idea with this changing DNA or transcribing DNA into RNA is, so we're using still the same language, so we're still using ribonucleic acid, but it's just slightly different, but in any case, so we're using the same language between DNA and transcribing it into RNA. And so what we're going to go through is briefly how that works and a bit of the terminology that you need to understand which strand of DNA. Remember, DNA is always double-stranded, and there's only one strand that we read. So, for example, in your computer labs, you were reading the coding strand of DNA when you brought up that sequence of your transcription factor, the TP63 gene. Okay, so you were reading it 5', so at the beginning, to 3' to the end, and you were reading in one orientation, and you were reading only one of the two strands. So the DNA is always double-stranded, so there would have been a complementary strand to that one that you were reading. But that is the one that you're reading to see what sequence your TP63 gene is, and that's actually the sequence that you end up needing to copy into RNA to finally make a protein from that. Again, how many people did functional proteins and genes? Okay, and so the other people would have done genetics as their, okay. So the idea, again, and I'll reintroduce this when we look at DNA sequence, is that when you're reading this coding region of DNA, so where your gene, ATGC, whatever your gene sequence is, every triplet codon of your gene sequence codes for a particular amino acid that then gets made in a line, and you finally make a protein, a functional protein from that. So it's very important which strand of DNA you copy or you transcribe into RNA to make sure that you're making the right strand of RNA to then finally make the protein that you actually want to make. Okay, so in this figure, so the DNA, the double-stranded DNA, you can see it's got two colours. So the coding strand, or the strand that we read, and now you've done your computer practice, it's easy to say, the one that you're reading your gene sequence from is in blue. And you can see it's the one that is, so the one that is reading 5' to 3' is the strand that you're reading off your gene sequence. The other strand is the red one, denoted as a red colour here. And you can see that's the reverse complement. So it's the complementary strand of that particular DNA sequence. And this strand is what's called the template strand. And we'll talk about this in more detail because that's the strand that we use to make our RNA, to make to transcribe DNA into RNA. The enzyme that does that is called RNA polymerase. So just remember, anytime you see A's on the end of a protein, it's an enzyme, it does something, okay? And so in this case, it's a polymerase, so it polymerizes, it pretty much joins up nucleic acids and makes a new DNA. In this case, it's an RNA polymerase, so it makes a new RNA strand. Okay, so let's have a, so now we have the same colour scheme. So the strand that we were looking at in blue here is the same strand in blue here. So this is the coding strand, the strand that we often refer to as the coding strand, which is the strand that we're reading the gene sequence on. It's also called the non-template strand. So that's why it's very important to get this nomenclature down packed so you actually know what we're talking about when we're talking about template strand, coding strand, non-coding strand, and so it doesn't become like a bit of a mess. Okay, so the coding strand of DNA, so the strand of DNA where did my mouse go? Where you're reading your gene sequence from, 5' to 3' is the blue strand. Every gene will start with a start codon. And you can see the start codon is always only one codon, so only one triplet codon of DNA, and it's ATG. Okay, so when we talk about annotating genomes, once they've been sequenced, obviously one of the main points of annotating a genome is to find the start and the end of all the genes that are in that organism. And we'll talk about annotating a genome in the next few slides. So for the purposes of this explanation, so we've got our coding strand of DNA. Here's our start of our gene, whatever that gene is, and there's the rest of the sequence. And we're reading it, in this case, from left to right, 5' at the beginning to 3' at the end. And you can see then the reverse complement, so the complementary strand of DNA, because DNA is double-stranded, is called the template strand, okay? And it's really important to understand that that is the complement to the strand that you're reading, so the coding, where your gene sequence is. And you can see it's it's a complement, okay? So G base pairs with C, A base pairs with T, etc., etc. And you can see if you keep on going along, you also make the complement of the start codon. So here, there's no more start codon in this sequence. We don't read it as a start codon, it's TAC. Okay, so if we were to make protein from that, we wouldn't find a start codon in that in this sequence. So if we would be reading this as our coding strand of DNA, there's no start codon here. We wouldn't make we wouldn't be transcribing that gene or the start of that gene sequence. We only need to, essentially not using this strand as the, we're using this strand as the way of making the new RNA strand, because how RNA polymerase works, it reads the template strand and it makes a complement of that template strand. So you can see then the new RNA strand is complementary to the red strand, to our template strand. So the RNA polymerase needs a template to start its polymerization of RNA, so to make a new RNA molecule, and it will read the complementary strand to our coding strand, so where our gene sequence is, and put the complementary base in. And what you can see then is once it does that, you now have an RNA strand that is the same as the blue strand. Okay, so it's the same as our coding strand or our non-template strand. And now we're reading exactly the same sequence. Obviously there's a few differences, or one difference in RNA. We have a different base. We have a uracil instead of a thymine. We'll get to that in a minute. But in essence, it's the same DNA, it's the same ribonucleotide sequence. You can see now we have an AUG or an ATG start codon. We can see that's the start of our gene. That's important because that singular RNA strand is what gets read by the translation machinery to finally make your protein. And so once you see, for example, the start codon, a start codon always codes for methionine. So an ATG codon always codes for methionine, it's always the start of a protein sequence. And then the next three codons along will code for another amino acid, and the next another amino acid, etc., until you make your whole new nascent protein polypeptide chain, and you can finally make your protein. So this concept of which one is the strand of DNA that we use to copy our RNA is important to understand. And it's not the strand that we read our gene sequence from. So it's not the coding strand of DNA, it's the complement. And the reason for that is because the enzyme that does this process needs a template. So it needs to be able to read a strand to then put the complementary base to make the new complementary base to it and make the new RNA strand. Okay, so this idea and this process is important, and you do have to understand the nomenclature of the different strands and which ones we're talking about when we're talking about transcription. So Mark is going to do transcription. He's going to deal with it in a lecture and regulation of transcription, so in a couple of lectures actually, and you'll get a lot more information on the enzyme or the RNA polymerase and the other enzymes involved in this process. So it will be a much more involved set of lectures when Mark talks about transcription. So this is, again, to introduce this idea of the nomenclature of which strand gets copied into which strand of the DNA gets used to make this new RNA strand, which is our intermediary until we finally translate that RNA into a protein. Okay, so we've been talking about, so here when we're talking about transcription, so we're talking about a messenger RNA molecule that finally gets made into a protein. There are also other types of RNA molecules that don't get made into proteins, and you'll be talking about those in the context of when they're used. they're called transfer RNA or tRNA, and ribosomal RNA or rRNA. So these two types of RNAs, so they get made into RNA by a specific RNA polymerase, but they get used as RNA. Okay, so they don't get made, these types of RNAs don't get made into functional proteins because they have a function as RNA, and they both have functions in translation, so in making that messenger RNA into a protein. So you can see RNA and the differences here in the different types of RNA is you have different RNA polymerase enzymes that deal with making those different types of RNA. So Mark will talk about RNA polymerase 2 and its features, and this is the main RNA that makes this messenger RNA that we finally make into a protein. And you can see there's RNA polymerase 1 and 3 that deal with the other what we call non-coding RNAs. So ones that stay as RNA, as a functional RNA molecule, and have a role in translation, which Mark will also talk about, which is what's on that slide. Okay, so this concept of the template strand being the complementary strand to the strand you're reading the gene sequence of, that's the strand that the RNA polymerase uses as a template to copy the RNA, the new RNA, and make essentially a copy of the blue strand. Okay, so that's what we want in the end. If we copied the red strand, we would not get the same sequence. Okay, so it's very important to understand this concept. Okay, so does anyone want me to explain this again? Because this is really a crucial thing to then understanding the whole process of transcription and how, yeah. Yeah, so I'll come to that when I've got a slide about the RNA polymerase and how it reads. But yeah, we'll come back to that. Okay, so what I wanted to do was to just introduce the molecules, RNA and DNA, and the fact that they're pretty similar, and just the few differences that there are between RNA and DNA. So remember, the DNA molecule and the RNA molecule, they both have a phosphodiester backbone. And then there are the DNA or the RNA bases hanging off that ribose sugar. And we've got two types of DNA or RNA bases, and they're either a double ring structure or a single ring structure. And they're called different, I have another slide of that further down. Okay, so this idea about the RNA polymerase and which way the new RNA is transcribed. So the RNA polymerase actually reads the DNA, the template DNA, essentially in the opposite direction, but it synthesizes the DNA 5' to 3'. So it's essentially it reads. So it needs the template to do that, and it reads the template 3'. So this is our template DNA strand now. So sorry, it's now changed from side to side, to up and down. So it reads the template DNA strand 3' to 5', and it reads whatever DNA base is there, and it puts on the complementary base. And the growth of the strand is in the 5' to 3' direction. And that happens all the way down until it finds the stop codon of that gene. Okay, so what are the differences then between this DNA and RNA? So as I said, this transcription event is a copying of DNA into the RNA molecule, and so it's nearly a total copy. The only difference is that the RNA has, rather than a thymine, it has a uracil in the in its strand. So a thymine and a uracil are nearly the same. It's just this methyl group here at the 5' position of the ring that's different. Okay, so that's essentially the only difference between RNA and DNA. So DNA gets its name because it's deoxy, so ribose sugar. So is there something different in the sugar of this RNA and DNA that you can see? That's the other main difference between DNA and RNA. Any difference? It's in red. I can't hear anything. Someone said something?
No oxygen.
Yeah, deoxy. Anytime you see D something, it means something's gone from something. So it's so in this case it's deoxy. There's oxygen is gone. That's how the DNA gets its name. And you can see at this 2' position in the sugar ring, in the ribose ring, is no hydroxyl anymore. Okay, so it's just a hydrogen. And in the ribose sugar, then clearly that's still a hydroxyl group. So it's still an OH group. And that's you can see that DNA and RNA are pretty much very, very similar, and that's why this idea of transcribing, of copying DNA into RNA using the same language. So it's still the same language of molecules being used. Okay, so we'll just go through, because not everyone did functional proteins and genes. We did talk about building DNA and RNA in that unit, but for the people who haven't done that, we'll just rehash the nomenclature of the DNA and RNA molecules. Okay, so remember we've got a double ring structure that is a DNA or RNA base. Actually, only DNA bases, adenine and guanine. They're called purines, and the single ring structures have the longer name. They're called pyrimidines, and uracil is only in RNA and takes the place of thymine in RNA. Okay, so the basic structure is we've got a DNA base, a ribose ring, a 5-membered sugar, and a phosphate group, and that's essentially the phosphate and the sugar make up the RNA or DNA backbone, and then the different bases get put on the ends of either RNA or DNA in this way. So we've got, again, the sugar ring, phosphate, and then whatever DNA or RNA base we need to have there. Okay, so a little bit of nomenclature. When you see these things, you know what we're talking about. So again, the DNA RNA base, we've just talked about that. Clearly in DNA, then they base pair, and we only ever see a base pair between a and a T and a G and a C, and we'll come back to that in a minute. And the sugar, we're always talking about the ribose ring or the pentose ring. The nucleotide is when we're talking about all three, so we're talking about the DNA base, the sugar and the phosphate, and so this is now the sugar-phosphate backbone that is attached and to make the DNA or RNA backbone. When we're talking about just the sugar and the DNA and the phosphate group, this is referred to as a nucleoside. And you will see both of these, nucleotide and nucleoside, being used, and it's useful to know what it is that they're talking about, which part of the structure it is that they're talking about. And this is just an example of when you of how you can talk about this. So this is adenosine. So the adenosine base. We're talking about the adenosine nucleoside, we're essentially only talking about the DNA base and the sugar, and then we're talking about the nucleotide once we start talking about phosphates being added on, and you can see these are the different molecules that that adenosine nucleotide can make with phosphate. So you can either have a 1-phosphate group attached, 2 or 3. And everyone should know about ATP, which is the molecule that essentially we use as an energy transaction in the cell. Okay, so the DNA is a double helical structure. So this sugar phosphate backbone that we've been talking about, it actually forms, it's on the outside of the helix. And the DNA bases that base pair together form the inside of this DNA double helix. And so remember that one strand goes in one direction and the other strand goes in the other direction. So they're anti-parallel. The other thing that's very important to remember when you have DNA base pairing, again, the sugar phosphate backbone is always on the outside of this double-stranded helix, and this base pairing is occurring between essentially a double ring structure and a single ring structure. Okay, so you can never have, it's geometrical, and we'll have a look at the fact that it's not quite like an alpha helix in a protein, so it's got a slightly different geometry, but you can only ever have a purine base pairing with a pyrimidine. So a double ring structure can only have a base pair with a single ring structure, because or else you won't continue this perfect geometry of the DNA double helix. So adenine always base pairs with thymine. It's important to note they make 2 hydrogen bonds when they base pair, and guanine always base pairs with cytosine, and they make they have an extra hydrogen bond, and this becomes important when we start talking about breaking open the DNA strand to be able to do anything with it, and we'll talk about that in more detail. Okay, so so as I said, that the DNA double helix, it's geometrical, so it's the same. It continues this conserved double helical structure all the way down, but it's not like a spring. Okay, so it's not quite. It's not. There are differences in the sides of the DNA double helix, and essentially what that means is that there is, for example, so the DNA double helix has, as the 2 helices wind their way around, you can see it's got a larger gap and then a smaller gap, and then it would have a larger gap and then a smaller gap, and they're called the major groove and the minor groove of DNA. Okay, and so that is repeated all the way down the DNA double helix. And these major groove and minor grooves are really important for protein DNA interactions, and they're the areas where the regulatory proteins like transcription factors that we'll be talking about subsequently bind the DNA and help turn on, for example, expression of a gene. Different base pairs can affect the structure of DNA, and for example, if you have a bit of the DNA strand that is rich in AT base pairs, remember this AT base pair only have 2 hydrogen bonds holding it together, and that's a region that can be opened much easier in the DNA, and we're going to be talking about that when we start talking about promoter regions, so these areas of the DNA before the gene sequence starts that we have to get the transcription factor machinery onto to finally transcribe our gene. And these areas of the DNA are really important to essentially open up the DNA. So you have to remember the DNA double helix, it really is a storage form of DNA. You can't do anything with the DNA when it is in its double helical form. Okay, you need to open up the DNA to be able to do anything with it. And you can see you needed to do that to transcribe the DNA into RNA. You need to do that if you need to replicate the DNA. You need to do that if you have to repair the DNA. Okay, so the double helix is essentially just the storage form it is the storage form of all your genetic information, and anytime you need to do anything with the DNA, you need to be able to break it open. Okay, so what DNA carries important information. So we clearly it carries this structural information, these areas of the DNA double helix that are conserved and that DNA binding proteins bind to, and we'll come and talk about that in subsequent lectures. And then there's also the sequence information. So the specific arrangement of DNA base pairs and regions of the DNA that are conserved and that help, for example, conserved, so the actual gene sequence that will be conserved, but also these areas of the DNA before and after that gene that help regulate whether we're turning on a gene on and off. And we'll be talking about that in a lot more detail in subsequent lectures. Okay, so the sequence of DNA determines its chemical and thermodynamic properties. Like I said, if you have a lot of A's and T's base pairing in your DNA strand, that DNA strand can get pulled open much easier. If you have lots of G's and C's, they've got 3 hydrogen bonds. You need a lot more thermodynamic energy, heat essentially, to open up those strands, and that's important as well, and we'll talk about that in the context of, for example, the polymerase chain reaction. The DNA base sequence is important because it also provides recognition sequences for enzymes that cut the DNA. So you saw a nice example of this BTS1 in your computer prac. So it's a restriction enzyme, has a specific site where when it sees that site on the DNA, it makes a cut. Okay, and so the base sequence provides these type of recognition sequences for restriction endonucleases. And then also the idea of the fact that specific bases or stretches of bases can be very highly conserved if they're required, if they're part of the gene sequence or if they're required for that gene's function. Okay, so we actually talked about this already, but we'll talk about this a little bit more in the context of opening up the DNA, because that's essentially the idea of the polymerase chain reaction is for us to do anything with DNA, we need to open it up into its single strands to then make a copy of it. Okay, so the DNA polymerase is the same enzyme that you'll see when you replicate, when a cell replicates its DNA. So we've, again, taken this idea of a molecular biology tool, but the only way that we know that we can use that tool in that way is because we understand how replication occurs in a cell, and then we can use that enzyme as a tool in vitro in the tube. Okay, so the numbers of A's and T base pairs or G and C base pairs in your DNA sequence, say in your gene sequence that you're amplifying up, you can calculate what's called a melting temperature for that region of DNA, and it's basically related to the number of G and C and A and T base pairs that you have in that sequence. And generally, the more G's and C base pairs you have, the higher the temperature you require to, or the higher thermodynamic force you need to open up that piece of DNA. Okay, so that's the idea then of the polymerase chain reaction, but of anything that requires the DNA to be in its single strands. So when we replicate the DNA, when we transcribe the DNA into RNA, when we repair the DNA, we have to go through this separation process of the DNA strand. So separating the two strands is called denaturation. So this is the kind of terminology you'll see over and over again when we're talking about the polymerase chain reaction, but that's occurring also obviously in vivo in cells to open up the DNA. So this is where we're opening up the DNA. That's what denaturing means. In the tube, we do that by heat. Okay, so we heat up the reaction to 95 degrees and the DNA strands come apart. Annealing is the process by which, when you have two complementary strands, they stick back again. Okay, so you might heat them up in the tube, but if they're complementary, they'll stick back down again. Okay, and so that's the process of annealing. That's also the process that is used when you use these little 4 pieces of DNA, these forward and reverse primers that you found for your TP63 gene sequence. They're going to anneal to those regions of your TP63 gene when you do your first polymerase chain reaction. And we often talk about melting point of the primers, the primers that you use. And the melting point is essentially the temperature at which half of your molecule is double-stranded and half is single-stranded. And you can work these things out for each sequence that you use. And it's important to know that when you set up a polymerase chain reaction, the actual machine that you use, and you won't be setting it up, it will already be set up for you, but the temperatures that we use for annealing are dependent on the melting point or the melting temperature of those 2 small oligonucleotides that we use. Okay, so the last few slides of this lecture, I'm going to just introduce this idea of the fact that there's a lot of information in the DNA. If you were to have it in a line, for example, the human genome would be 2 meters long, okay? But every cell in your body, or well, we're talking about humans now, but every cell in your body has that 2 meter long bit of DNA that has to fit inside the nucleus of the cell. Okay, so something has to happen to the DNA to be able to fit in your microscopic cell, each cell of your body with a nucleus with that 2 meter long DNA. So clearly it doesn't stay as a chain, as a DNA double helix. Something has to happen to that DNA, must be severely compacted for it to be able to fit into every cell of your body. Okay, so what happens? So this process of compaction is a highly regulated and controlled process. Mark will spend a lecture talking about the process in more detail, but I'm going to introduce it here just to get you thinking about the fact that the DNA, again, this storage form of genetic information, gets compacted up into chromosomes, but we need to access that DNA if we need to transcribe that DNA into RNA, and we need to translate that RNA into the language of amino acids to finally make our protein. So something has to happen in this information flow that is very ordered for us to be able to access our storage form of genetic information. Okay, so what happens when we have to compact the DNA is that the DNA starts to associate with some proteins, and we'll briefly talk about which ones they are. Again, Mark will talk about them more in his lectures. When we're talking about genomic DNA associating with these proteins, we refer to the DNA as chromatin. Okay, so when the DNA is free, it's referred to as genomic DNA. When the DNA is starting to be compacted up into these histone proteins, which is what they're called, we refer to the DNA as chromatinized DNA or chromatin. And the most common proteins that associate with genomic DNA are called histones. And I'll show you a very nice crystal structure of histone proteins wrapping DNA and compacting DNA in the next slide. So this slide I just put up just to remind you of this fact that the chromosomes are essentially our storage form. This is where our DNA is compacted up and stored as chromosomes. But for us to replicate the genome, so each cell has to replicate, this DNA must be uncompacted for you to be able to replicate your DNA. And you can actually see this under the microscope. You can see this decondensing, so condensed chromatin, so the very compact form of DNA bound to these histone proteins, and then decondensing, so uncompacting the DNA. You can actually see that when you look at the different phases of the cell cycle. So it's a very dynamic process that's occurring in cells all the time to be able to organise the DNA for it to be compacted and decompacted depending on whether it's needed or needs to be replicated or a gene needs to be made into a protein, etc. Okay, so the idea is that DNA is wrapped around these histone proteins, and Mark will talk a bit more about the fact that each one of these histone proteins, there's 8 of them, it makes what's called a nucleosome, and you can see essentially DNA being wrapped around these histone proteins, and they make beads on a string. So if you were to see this under an electron microscope, you would see this image. So you would see what looks like beads on a string. And you can see there are some very nice, more recent crystal structures of these histone proteins wrapping DNA and showing the first step in compaction of the DNA. And again, we wouldn't understand how DNA got compacted in the first place if you don't see these 3-dimensional structures in atomic detail. You would have no idea how this compaction initiates. And so this is the first step in DNA compaction. So you can see DNA is wrapped around the outside of these histone proteins, and the DNA is compacted in 6 to 7-fold. So this is the first step of DNA compaction that occurs. And that's not just the only thing that occurs. We have to get DNA to be from this double helical structure to a chromosome. So there's all these other steps of compaction, further compaction that occurs. So we've got these beads on a string, which is essentially the first step of compaction. We then make a filament. We then make an extended form of the chromosome. We then make a condensed chromosome, and finally this mitotic chromosome, this chromosome that everyone knows what a chromosome looks like, that is the final compacted version of the DNA. And so for us to do anything in the cell, we have to decompact the DNA before you can do anything. So before you can replicate the DNA, transcribe a gene, repair a piece of DNA that is damaged. So you can see that there's lots of stuff that has to occur for you to be able to access your genetic information to do anything with the DNA to start off with. And so it's like an amazing process that has to occur for you to access your DNA in the first place. Okay, and so that's where I'm going to leave it for today, and then next week we'll start off talking a little bit more about genomes and annotating genomes.